The main contribution of our paper is that we have explored a precise and fine landscape of amyloid beta pathology of the Alzheimer's disease brain with the emerging technology of the MALDI mass spectrometry. We achieved technological advancements by generating a special protocol with formic acid pretreatment of the tissue slides.
Following MALDI-IMS, segmentation maps are generated and calculations are performed to discover novel marker proteins or peptides co-localized with plaque and subarachnoid vasculature.
Obtain human cortical specimens for IMS from brains that were removed, processed, and stored at -80 degrees Celsius within 8 hours post-mortem. Take the brain specimen from the occipital cortex of AD patients in age-matched controls. To cut the tissue sections on a cryostat, first place conductive indium tin oxide or ITO coated microscope glass slides inside the cryostat. Warm the autopsy brain specimen from -80 degrees Celsius to -22 degrees Celsius inside the cryostat. Attach a new disposable blade to the cryostat for every experiment. Always try to use a clean part of the blade. Put the frozen autopsy brains on the stage along with a small amount of optimal cutting temperature compound. For IMS and immunohistochemistry, cut 5 to 6 sections from each tissue sample. When the blade is just starting to cut the tissue, turn the wheel and face the block until all of the